Have you ever stopped to think about how truly wild it is that every single cell in your body, a neuron in your brain, a cell in your skin, is built from the exact same genetic blueprint? It's the same book, but it tells completely different stories. And the key to that incredible magic lies in a process called transcription.
So that's the big puzzle, right? If every cell has the same DNA, the same book of life, how does one cell know to read only the chapter on how to be a neuron, while another cell only reads the chapter on how to be a skin cell? That process, controlling which chapters get read and when, is exactly what we're going to unpack.
At its heart, transcription is a really elegant idea. Imagine your DNA is this massive, priceless library of architectural blueprints. It's too valuable to take out of the library and bring to a construction site. So what do? You use a master photocopier to make a working copy. That's the RNA. That copy can then leave the library and be used to actually build things, in this case, protein.
But, our first real understanding of this process didn't come from studying our own complex cells. Nope, it came from bacteria. And as scientists were about to find out, reading the simple little instruction manual of a bacterium is a whole different world from navigating the massive, sprawling library inside each one of us.
And when you put them side by side, the difference is just staggering. In bacteria, you basically got a simple manual. There's one main photocopier, this RNA polymerase. The on-off switches are pretty straightforward, and the DNA itself is just kind of floating around, super easy to get to. But in our cells, it's a vast library. We don't have one photocopier, we have multiple specialized ones. The switches are this unbelievably complex web of regulation, and our DNA blueprints are carefully packed and wound up, making them way harder to access. This complexity was the first big clue that our story was going to be a lot more involved.
For a long time, that simple bacterial model was pretty much all scientists had to go on. But then 1969 happened, and that simple universe of understanding was completely shattered by what biologist Robert G. Roeder later called the Big Bang of this field, a single discovery that absolutely changed everything.
And you can see just how fast things changed on this timeline. In 1961, we had this neat, tidy model from bacteria, one machine, simple rules. Then, boom, 1969, Roeder finds out that our cells don't have just one, but 3 totally different RNA polymerases. That immediately told us there was a whole new layer of control going on. And then, through the 70s, the mystery got even deeper when scientists figured out that these 3 fancy machines couldn't even start the job on their own. They needed help.
And you can just feel the excitement of that moment in Roeder's own words. This wasn't just some small finding. For him, it was a true eureka moment. The kind that not only makes your career, but also just blows your mind with a million new questions and possibilities.
And get this, here's a little bit of science history that I just love. That huge, field-changing paper that was published in the journal Nature, it was rejected at first. The reviewers actually said it wasn't of general interest. It's just a perfect reminder that sometimes, even the most revolutionary ideas take a minute to sink in.
So what do these 3 different photocopiers actually do? Well, think of them as a specialized team. Polymerase I is the factory worker, cranking out parts for the cell's protein factories, the ribosomes. Polymerase II is the one we hear about most often. It's the master architect, making the messenger RNA, which are the actual blueprints for our proteins. And polymerase III is kind of the logistics expert, making things like transfer RNAs, the delivery trucks that bring supplies, and other small essential utility molecules.
Okay, so having 3 polymerases was a huge step. It explained how our cells could control making different kinds of RNA. But it also opened up a brand new mystery. When scientists tried to get these polymerases to work in a test tube with just pure DNA, nothing happened. It became obvious pretty quickly that these sophisticated machines were basically useless without a whole crew of helper proteins to get them set up and ready to go.
And that brings us to this next incredible level of complexity, assembling the machine. See, it's not enough to just have the photocopier. You need a whole team to find the right blueprint in the library, load the paper, and hit the start button. It's less like a machine and a lot more like conducting a giant, complicated genetic orchestra.
The core members of this orchestra crew are called general transcription factors, or GTFs for short. The best way to think of them is like the essential stagehands for the whole operation. Their job is to find a gene's starting line, the promoter, build a stable platform, and then guide the main star, the RNA polymerase, right into position. Without them, the polymerase would just be floating around, totally lost.
So how does this all come together? Well, first, a site scout called TFID comes in and lands on a specific sequence, acting like a beacon. Then other factors swoop in to build and stabilize the platform,
Only then is the main photocopier RNA polymerase II brought to the site. After that, another crucial factor acts like a power switch, using energy to pry open the two DNA strands. And finally, liftoff. The polymerase breaks free and starts zipping down the DNA, making its RNA copy.
But that's just the basic crew that can assemble at any gene. To choose a specific gene, to decide which blueprint to copy right now, you need another layer of management. These are the activators. Think of them as project managers. They bind to DNA way far away from the gene and send out the signal, "We need this one." And to get that signal to the crew at the start line, you have cofactors. They act like foremen, physically looping the DNA around to connect the distant manager to the machinery, telling them, "Okay, team, this is the one we're copying."
And you know, the discovery of the very first one of these project managers, a protein called TFIIA, was another one of those game-changing moments. It was the first hard proof that to turn on a specific gene in a complex organism, you needed a specific protein to kick the whole process off. It basically established the central rule of the game for how our cells get so precise.
So, we've gone from one simple polymerase to three, from a single machine to this massive, dynamic orchestra of proteins. The picture has gotten so much richer. But the wild part is, the universe of transcription is still expanding. Let's take a quick look at the absolute cutting edge of what we're learning today.
I mean, we are now diving into whole new layers of control we barely knew existed. We found things called super enhancers, which are like these giant regulatory motherboards that control the most important genes. We're also realizing that the 3D shape of our genome is absolutely critical. It folds up into these neighborhoods that decide which genes can even talk to each other. And maybe the weirdest of all, we're finding that these proteins can actually form tiny liquid-like droplets, basically creating little pop-up factories inside the nucleus to make transcription incredibly efficient. It's just amazing.
So, we've taken this journey from a simple little photocopier in bacteria to a vast three-dimensional dynamic universe of regulation humming away inside every one of our cells. And the more we map this incredible universe, the more we realize just how much is left to explore. It really just leaves you with that one big exciting question that drives all of science. What fundamental new rules of life are still out there, just waiting for us to discover them?